Thanks everyone for your patience while we resolve some technical issues. I'm really excited to be here today. I'm Shauna Clark, and I'm the Segment Marketing Manager at Oxford Nanopore Technologies focused on single-cell sequencing applications. I'm delighted to welcome you all to this knowledge exchange, where we'll be hearing from Rebecca Pollock and Neil Horner about implementing Oxford Nanopore's single-cell sequencing workflow. After their talks, they will be taking part in a live Q&A, so please add your questions to the Q&A box, which you'll find at the bottom of your screen. You can also upvote questions you would like most answered, and we'll try to get through as many as we can. This knowledge exchange is being recorded and will be made available on demand after the broadcast. But before I hand it over to Rebecca and Neil, I'd just like to provide a brief introduction to single-cell sequencing with Oxford Nanopore. All right, let's get started now with a brief introduction. Over the past 5 years, single-cell and spatial methods have revolutionized transcriptomics. Now researchers are, at a massive scale, able to look at the unit of biology, the cell. And this transformative technology has been featured in Science and Nature Methods. Researchers are now able to make discoveries of new cell types they didn't even know existed. Dr. Aviv Regev and team was able to discover the ionocyte, a new cell type found in the lung. This cell type was implicated in cystic fibrosis as it actually expressed the CFTR gene at a very high level, even though it was very rare. This has implications for how researchers can think about treating this disease. Despite the discoveries being made, most of this work has been done with short-read-only technology. And short reads miss about 80% of most transcripts. And the image shown here at the top are the annotation of the transcripts from a gene. In the center are the short reads aligning to the transcript. And the bottom panel shows Oxford Nanopore reads aligning to the full-length transcript. So why is this missing biology important? To read the quotes on the side here, up to 95% of human multi-exon genes undergo alternative splicing. And short-read sequencing leads to a loss of valuable transcript information, such as genetic variants that can only be derived from an intact cDNA molecule. And some of those variants include gene fusions, SNVs, and also transcripts for nonsense-mediated decay. So how do we recover this ultra-rich biology that we're missing with short reads? You can go beyond gene counting with long nanopore reads. You can get more biology with gene and isoform expression plus variant detection in a single experiment. We also offer simple data analysis with our Epitome workflow, which includes cell barcode and UMI demultiplexing without the aid of short reads. We also have a very efficient workflow. You can go from cDNA to sequencing-ready library in about 3 hours, and you can also use existing 10x genomic cDNA that is probably in your freezer right now. The data on the right is showing an example of how isoform level detect gene expression is important. You can see that the isoform expressed during neuronal development changes from the 204 isoform is highly expressed in mature cells, while the 206 isoform is highly expressed in precursor cells. This change in isoform level expression would be missed by using a short read only technology. And now I'm going to move on to a quick overview of our workflow. We offer fully supported end-to-end protocols for the 10x Genomics Chromium NextGem single-cell 3' and 5' kits, and Rebecca will dive deeper into those details coming up next. We also have demonstrated compatibility with Visium spatial gene expression, as shown in our partner application note with 10x Genomics, which you can find on our website. Briefly, to prepare a nanopore library, you start with 10 nanograms of 10x Genomics cDNA and simply use our nanopore sequencing kit. We recommend sequencing on a PromethION device, as a single PromethION flow cell can provide about 80 million cell-assigned reads. After sequencing, we offer the workflow single-cell pipeline for your data analysis. And Neil will take you deeper into this, showing you how to set up the analysis as well as going through the results. So with that, I'm going to pass it off to Rebecca to take you on a deeper dive into the library preparation and nanopore sequencing section of the workflow.
Yeah, hi. So I will talk to you about the single cell workflow. So I'm mainly going to focus on library preparation right through to sequencing with our PromethION devices. My name's Rebecca. I'm a development scientist here at ONT, and I will be able to answer any questions you have on our protocols. So this is just an overview of what you might expect if you were running the 10x all the way through to sequencing with ONT. So firstly, it's very important that you have viable cells. So we want these to be preferably above 90% viable, and that's what 10x themselves recommend. So in our case, this was PBMCs from human blood. That we put 5,000 into the 10x Chromium X, and then we moved on through our protocols. So with the Chromium, you would expect to either run the single cell 3' kit or the 5' kit, and then it's important that we quantify and quality check that cDNA, because you'll only need 10 nanograms of that DNA to go into our ONT protocols. So the 3' protocol or the 5' protocol are both available on the community, and I'll talk you through those in detail. So this is an overview of those protocols, the 3' cDNA protocol and the 5' cDNA protocol. The 3' is a little bit longer than the 5', so this has two separate PCR steps. The first with biotinylated primers and their normal primers, and the 5' cDNA doesn't have those biotinylated primers, so you only have a short 8-cycle PCR step as opposed to 2. And then in both preparations, you'll go through to end prep, ligate the sequences for adaptive sequences, and then you'll be able to sequence them on a PromethION. So as I said before, these are both available, and in the future we will potentially release a VDJ protocol and a feature extension protocol to go along with these two protocols. So what would I need as a customer to run these protocols? As I said, there's a little bit more involved in the 3'. So for both, you would need an LSK114 kit. So this is the ligation sequencing kit. You might have heard of this before. Some of you might have even used it. It just has the adapters and everything ready for you to prepare libraries for sequencing. Specifically for the 3' cDNA, you would also need the PCR expansion. So this is for that second PCR that I mentioned. And then for both, you would need PromethION R10 flow cells, as many as you would need for your experiment. The other part to this is a set of custom ordered oligos, whether that's biotinylated or non-biotinylated, depending on the prep that you are going to run. So this is just to have a quick look at what you might see if you ran the 5' protocol and the cDNA amplicons that you might expect to see. So this is an example from the 5', but it would also be very similar with the 3' kit. And we were yielding about 600 nanograms per sample. So that's why I made a note about the 10 nanogram input, because as you'll see, you'll actually end up with a lot of DNA from your runs, and very little needs to go into the protocol. So it does give you a lot of scope in terms of replicates. Now, to go a little bit deeper on those protocols, I wanted to explain to you why the 3' protocol is longer, and also just to show you what the sequences look like when they come from the box all the way through to going through to sequencing. So the main difference between the 3' and the 5' is this initial sequence. So this is the sequence that is attached to the gem bead. So firstly, you'll see it has the read one, the barcode, and the unique molecular identifier before the sequence. And this means that to amplify that, we require a template switching oligo on the other end. Now, the only issue with this is when you amplify that DNA, you get template switching oligo artifacts. So you get sequences with TSO, TSO on either end, and these are quite long fragments. So not only does that influence the sequencing, but we don't want to sequence things that aren't proper transcripts. We don't want those in our preps. So the reason that we have two PCRs is that this biotin strep pull down removes that artifact, and then we can go on to amplify just the regions that we want to amplify. So once you've got through your second PCR and your cleanup, you can you have your cDNA ready to end prep with 200 femtomoles of cDNA to adapt, and then we recommend sequencing 30 to 50 femtomoles of that product, which will give you really good pore occupancy and some really nice reads. And then just to confirm and to show you the difference with the 5', so this is a shorter prep, but it's because the TSO is actually within the sequence here, so when it's amplified you don't end up with that artifact. So to amplify these sequences we actually just need the read one and then a partial non-poly DT primer, and then that can go straight into the end prep and ligation again to sequence that 30 to 50 femtomoles. So why is there different protocols? I think I've covered that mostly, but that's just to do with these TSO artifacts. And this just generally means that there's less hands-on time during the library prep, so it's just a little bit shorter. But just to confirm that you don't need those primers, and we have definitely spent a lot of time checking biotin pull down versus no pull down. So as you can see from this graph on the right, the difference between the full length reads you're going to get and the total tagged reads, so this is those with a barcode and a UMI, the difference is very, very minimal between them, if not completely the same, and this is actually what we also see with gene expression and the transcripts that you're able to get out at the other end, is that the difference between these protocols is very minimal, so we do recommend the shorter 5' protocol. So moving on to sequencing, I mentioned that we sequence on the PromethION. So if you are looking at the cell barcoded reads, so those with barcodes and UMIs, you're looking to expect around 100 million reads for the 3' protocol and 60 million reads for the 5' protocol. And we do have different options for the project. So whether it's a small through to a very large project, we have the P2 and the P2 solo. So these are two PromethION cells at any one time. But then we can scale that right up to a P24 and a P48. So if you have a lot of samples and a lot of flow cells that you wish to run, then we can go all the way up to 48 flow cells at once. And the parameters for the 3' and 5' sequencing runs. So obviously you will select the ligation sequencing kit, so that's LSK114 that I mentioned before, and we recommend a 72-hour run length. So this is just to make the absolute maximum number of reads that you can get and just to get the best output that you can get. We also recommend 200 base pair minimum read length, so this is just to cover all of the reads you can, because some of those are quite short, those cDNA lengths are quite short. And with regards to the model, we recommend Dorado, and we are now able to recommend HAC. So this is high accuracy base calling. It is a shorter time to generate those HAC sequences than it is to use SUP, but the performance is almost the same. But if you do really want a maximum read output, there's about a 5% difference between the two models. So you might want to run SUP if that's what you need. And lastly, I'm just going to show you a little bit of example data. So this is from cDNA with our 5000 ppmc's 95% viability prepared with the ONT 3' protocol. So as you can see, we get a really nice read distribution. The N50 is between 800 and 900 base pairs generally for the 3' samples. We also get very good pore occupancy and run time. So in total, you would be able to get above 130 million reads for the 3' kit and above 89 million reads for the 5'. So I hope that I've covered most of the aspects of the protocols that you'd be interested in, but again, please feel free to ask us any questions, and I'm going to hand you over to Neil, who's going to talk to you about the single cell workflow and the bioinformatics side of this and what you can do with that fantastic data.
Hi, I'm Neil Horner and I'm a software developer in the Customer Workflows group here at Oxford Nanopore. So thanks to Rebecca for talking about the wet lab side of things. I'm involved in the development of the data analysis workflow for analysing Oxford Nanopore single cell sequencing data and the workflow WF single cell. So WF single cell is part of the Epitome platform, which is developed here at Oxford Nanopore. It includes a collection of bioinformatics workflows geared towards ONT data and a desktop application for managing and running those workflows both locally and in the cloud. It comes pre-packaged with about 16 open source workflows, which are written in the bioinformatics framework Nextflow, which many of you will be familiar with. So no bioinformatics skills are needed and minimal software installations required to get up and running. And using the Epitome desktop is straightforward, as you'll see. So first of all, I'd like to just give a brief overview of how WF single cell works. So first off, you input some single cell FASTQ, and the workflow attempts to find adapters within the reads to find once it's found these adapters, it can strand orientate the reads so all the reads are in the same orientation, and it can identify full length reads, which is what we want, and it can split chimeric reads into sub segments. So the next step is to extract barcodes and UMIs, and once these are extracted, they're trimmed from the reads along with any adapters. And then the next step is to take those trimmed reads and align to the supplied reference genome. This alignment is then used by StringTie to generate a transcriptome of the bulk RNA, so all the transcripts in the sample from all cells are used to create a transcriptome. And then the reads are then assigned a gene and transcript by mapping the reads to this bulk transcriptome and applying some rules to then to apply the genes, to assign the genes and isoforms. The next step is to correct any errors in the barcodes and UMIs, and then once we've got the barcode and UMI information, it's possible to then create the expression matrices for gene expression and transcript expression, and then a workflow report and several other outputs are generated. And this can be seen in more detail if you follow the link at the bottom of the slide. So now I'd like to spend a bit of time on how to run workflow single cell. So the easiest way is to use the Epitome desktop application. So once the application is installed, fire up the application, click on View Workflows, and this will take you to a page where the available workflows are listed. If you don't see WF single cell in the installed section, just click on Available Workflows and you'll be able to install it easily from there. Once you go to the workflow overview page, there are several tasks that you can perform from there. You can run some demo data, which will, clicking on Use Demo Data will download a small test data set from the cloud and will run it to give you a flavor of what to expect. You can update the workflow from here and delete it if you wanted to. But we're going to click on Run this workflow, which will allow you to supply your own data. Clicking on that button will take you to the workflow setup page. So here you can supply all the options that are required to run the workflow. So the main options are the FASTQ path to the path to the FASTQ data, the 10x kit and version that you're using. This is important to specify correctly, and the number of cells that are expected. Each option has an info box above it that can be expanded, which will show you a description and some help text. And most of the options are usually fine with the default settings, apart from those that I've just mentioned. Once you're happy with the parameters, click on the Launch Workflow button and that will get things started. You'll see a progress window appear, which gives real-time feedback of how the workflow is progressing. Each one of these progress bars describes the progress of a next flow process within the workflow. And then once the workflow has completed, if you click on the report tab at the top right, you'll be able to view the workflow single cell report within the application, and from there you can also see some other next flow reports, which will give you information about technical information about how the workflow run, such as the time for each process and the amount of resources used for each process. If you don't want to or are not able to use the Epitome desktop application, for example, if you don't have permissions to install it, you can run all our workflows by the command line, and I'll just describe briefly how to do that. So there's the dependencies to install first. So these are Nextflow, Java, and Docker or Singularity. The command below shows how to run the workflow, and this will run version 1.10, which is the latest version. It will pull the latest version, that it will pull that version from our GitHub page, and you can see in the command we've added the options for the FASTQ path, the kit name, kit version, expected cells, the 10x reference genome directory, and the profile. So the profile just tells the workflow which container engine we want to run all our processes in. So by default, so if you leave that blank, the profile, it will attempt to use Docker. But if you want to use Singularity, you specify it there with the profile option. Okay, so now that WF single cell is completed, we should have some outputs that we can look at. So we'll go over some of those now. At this point, at the end of the workflow, we will have had we'll have some reads that are assigned a corrected barcode, so we can assign it to a cell, and it will have a corrected UMI, so we can assign it to an original mRNA molecule. So we only count each UMI once, and there will also have been assigned genes and transcripts, and from all this information we can generate gene and transcript expression matrices. And these row count matrices are then further processed, and this processing involves removing cells where that only have a few cells expressed. A few genes expressed. We also remove genes that are expressed in only a few cells, and removing cells that have a high proportion of reads that are of mitochondrial origin, which can be an indicator that the cell is dying. And then finally, the filter counts are normalized to 10,000 UMIs per cell, so that they can be compared between cells. The filtering parameters just described can be set in the workflow and are described in the green box. And both the row and processed matrices can be found in the user output directory. So another output of the workflow are UMAP plots. So these plots, so UMAP takes the high dimensional expression data in the processed expression matrices and projects this data into 2 dimensions so it can be visualized. It attempts to preserve the local and global structure so that groups of cells can be identified, such as different cell types or cells that are in different stages of the cell cycle, for example. So one use of this could be as a QC tool. So if you know your data set has a few different cell types in it that are usually easily distinguishable by gene expression, you could look at these plots to see if the same number of groups are identified, for example. The plot shown is a gene expression UMAP with expression data from peripheral blood mononuclear cells, or PBMCs. And this plot is annotated by a single gene expression, and in this case it's FTL. And we can see how the expression of FTL varies greatly between different groups of cells. You can, in the workflow, you can specify genes of interest to annotate plots by. So what you would do is add your list of gene names to a text file, and you point to that path using the UMAP plot genes parameter. If you want to run the UMAPs, you need to specify the plot UMAPs parameter. Plot UMAPs equals true in the command line, and you should know that UMAP algorithm is stochastic, so running it multiple times with the same parameters can lead to different results. So in order to have a bit of confidence in the results that you're seeing, the workflow runs the UMAPs several times, and the number of times it's run can be specified with the UMAP n repeats parameter. So we also the workflow also outputs UMAPs that are annotated by the total mitochondrial expression of each cell, and this can be used to identify potentially groups of cells that are dead or dying. So in the output directory, you'll also find, amongst other things, a BAM alignment file from the genomic alignment, and these alignments are tagged with the corrected barcode, UMI, and the gene and transcript that have been assigned to the read. There's a per read TSV file with the same information and some alignment information. There's a mitochondrial gene expression level per cell file, and also a transcriptome FASTA and annotation file that was generated during the isoform assignment stage. So one of the main outputs from the workflow is the workflow report, which is a HTML file that can be opened in any web browser. So at the start of the workflow report, we see our sequencing summary, which gives an idea of the read quality and read length distributions of your data. There's a table which contains basic information about the single cell experiment. And then we come to the adapter configuration summary. So if in the top right box we see a read structure, a labeled read structure, and this one would be a full length read. It's a read segment which is flanked by the 2 expected adapters in the expected orientation. But below that, in the example configurations, we can see that sometimes we see different configurations, such as read segments with only one adapter, or the wrong types of adapters, so 2 adapter 1s instead of an adapter 1 and an adapter 2. And the plot on the left summarizes the amount, the proportions of the different read configurations that we find in the sample. And in this example, most of the reads are classed as full length, which is good. Next, we have a plot which summarizes the proportions of reads that are assigned different information. So we see the numbers, the proportions of reads that are assigned as full length, the proportions of reads that are assigned that says total tagged in this instance. Total tagged just means the number of reads that were able to be assigned a valid barcode. And then we've got the proportion of reads, of total reads, that were able to be assigned a gene and a transcript isoform. Then we've got some diagnostic plots in the report. The first is the knee plot, and this is a common plot that you'll see in single cell experiments, and this serves as a visualization of the barcode filtering process. So in any single cell prep, there'll be cells with low read count, which will be noisy and you'll want to remove from your analysis. In the plot, we can see the vertical line where the filtering occurs. So on the x-axis, we have the cells are ranked by the number of reads, and the y-axis is the read count per cell. So the workflow tries to find the inflection point of the knee. This is the point where read counts suddenly drop off, and it's usual to find low quality cells to the right of this point. And at the top of the plot, we can see how many cells have been identified with this threshold. We also have the sequencing saturation plots. So on the top two plots, on the x-axis, we have the median reads per cell, and on the y-axis we either have the median genes found per cell or the median UMIs per cell. And this data is generated by taking the final data and subsampling it. And what this gives you an idea of, what this gives you an idea of is the amount of complexity in the library that has been captured. So if we'd captured much of the gene complexity of the cell, we'd expect the curve to flatten out as the median reads per cell increases. And in this case, we don't see that. So what that tells you is if it's likely that if you sequenced, if we got more sequencing data from this experiment, we'd likely find more genes, and the same with the UMIs as well. The plot on the bottom is a sequencing saturation plot, and this just shows you the fraction of UMIs that are non-unique. So in this case, 20% of the reads in the experiment, 20% of the UMIs were found more than once. So this also indicates that by increasing the depth of coverage, more information could be gathered from this experiment. So now we've taken a look at workflow single cell briefly. So in the next slides, we'll just have a look at some third-party tools that can also be used to analyze your single cell data. So the tools on this page work similarly to workflow single cell, and some have functionality that go beyond what workflow single cell can currently do. So Blaze and FlexiPlex, they both offer barcode and UMI extraction. FlexiPlex also will split chimeric reads. If you want to identify mutations in your single cell data, SC Nano GPS can do this. This can be useful in cancer cell analysis. Scicelo 2.1, as well as single cell support, can also has support for Visium spatial transcriptomics. And next are a couple of tools that you can use to explore and further analyze your expression matrices that come out of WF single cell. So the first is Surat, which is really commonly used in the single cell community. It's an R package, so this means you'll need to use it by the command line. And for users that would rather not use a command line, there are several tools, but one of these is Cell by Gene, and this offers an interactive desktop tool for exploring and analysing your single cell data. So that's it for me for now. Thank you for listening, and I'll hand you back.
Thank you, Rebecca and Neil, for those fantastic presentations. Well, I'm afraid that's all the time we have for today. Thank you once again to Neil and Rebecca for sharing their fantastic work and taking part in the Q&A. And so that leaves me to say a final thank you for joining us today. But before I go, I'm excited to share a first look at the innovative single-cell research being presented at London Calling 2024, taking place May 21st to May 24th. Featured single-cell topics include brain development, cell atlasing, and isoform analysis. We'll also be celebrating 10 years of MinION and look forward to seeing you there. You can register to join us online or in person using the QR code here. And if you have any further questions about your projects or getting started with nanopore sequencing, please contact support@nanoporetech.com. We would really appreciate your feedback on this knowledge exchange. Please head below to the polls tab next to the Q&A tab, where you will find a quick survey on how you found today's broadcast. This will remain open once the knowledge exchange ends. Thank you all, and enjoy the rest of your day.